In eukaryotic cells, an aggresome refers to an aggregation of misfolded proteins in the cell, formed when the protein degradation system of the cell is overwhelmed. Aggresome formation is a highly regulated process which possibly serves to organize misfolded proteins into a single location. Biogenesis. Correct folding requires proteins to assume one particular structure from a constellation of possible but incorrect conformations. The failure of polypeptides to adopt their proper structure is a major threat to cell function and viability. Consequently, elaborate systems have evolved to protect cells from the deleterious effects of misfolded proteins. Cells mainly deploy three mechanisms to counteract misfolded proteins: upregulating chaperones to assist protein refolding, proteolytic degradation of the misfolded, damaged proteins involving ubiquitin proteasome and autophagy lysosome systems, and formation of detergent-insoluble aggresomes by transporting the misfolded proteins along microtubules to a region near the nucleus. Intracellular deposition of misfolded protein aggregates into ubiquitin-rich cytoplasmic inclusions is linked to the pathogenesis of many diseases. Functional blockade of either degradative system leads to an enhanced aggresome formation. Why these aggregates form despite the existence of cellular machinery to recognize and degrade misfolded protein, and how they are delivered to cytoplasmic inclusions, are not known. Aggresome formation is accompanied by redistribution of the intermediate filament protein vimentin to form a cage surrounding a pericentrolar core of aggregated, ubiquitinated protein. Disruption of microtubules blocks the formation of aggresomes. Similarly, inhibition of proteasome function also prevents the degradation of unassembled presenilin-1, PSE1 molecules, leading to their aggregation and deposition in aggresomes. Aggresome formation is a general response of cells which occurs when the capacity of the proteasome is exceeded by the production of aggregation-prone misfolded proteins. Typically, an aggresome forms in response to a cellular stress which generates a large amount of misfolded or partially denatured protein. Hypothermia, overexpression of an insoluble or mutant protein, etc. The formation of the aggresome is largely believed to be a protective response, sequestering potentially cytotoxic aggregates and also acting as a staging center for eventual autophagic clearance from the cell. An aggresome forms around the microtubule organizing center in eukaryotic cells, adjacent to or enveloping the cell's centrosomes. Polyubiquitination tags the protein for retrograde transport via HDAC6 binding and microtubule-based motor protein, dynein. Moreover, substrates can also be targeted to the aggresome by a ubiquitin-independent pathway mediated by the stress-induced co-chaperone BAG3. BCL2-associated athanogene 3, which transfers misfolded protein substrates bound to HSP70, heat shock protein 70, directly onto the microtubule motor dynein. The protein aggregate is then transported along the microtubule and unloaded via ATPase P97, forming the aggresome. Mediators such as P62 are believed to be involved in aggresome formation in sequestering omegasomes, which bind and increase the size of the aggresome. The aggresome is eventually targeted for autophagic clearance for the cell. Some pathological proteins, such as alpha-synuclein, cannot be degraded and cause the aggresomes to form inclusion bodies. In Parkinson's disease, Lewy bodies, which contribute to neuronal dysfunction and death, triggering aggresome formation. Abnormal polypeptides that escape proteasome-dependent degradation and aggregate in cytosol can be transported via microtubules to an agressome, a recently discovered organelle where aggregated proteins are stored or degraded by autophagy. Synphilin 1, a protein implicated in Parkinson's disease, was used as a model to study mechanisms of aggresome formation. When expressed in naive HEK293 cells, synphilin 1 forms multiple, small, highly mobile aggregates. However, proteasome or HSP90 inhibition rapidly triggered their translocation into the aggressome. And surprisingly, this response was independent on the expression level of synphilin 1. Therefore, aggressome formation, but not aggregation of synphilin 1, represents a special cellular response to a failure of the proteasome chaperone machinery. Importantly, translocation to aggressomes required a special aggressome targeting signal within the sequence of synphilin 1, an ankyrin-like repeat domain. On the other hand, formation of multiple small aggregates required an entirely different segment within synphilin 1, indicating that aggregation and aggressome formation determinants can be separated genetically. Furthermore, substitution of the ankyrin-like repeat in synphilin 1 with an aggressome targeting signal from Huntington was sufficient for aggressome formation upon inhibition of the proteasome. Analogously, attachment of the ankyrin-like repeat to a Huntington fragment lacking its aggressome targeting signal promoted its transport to aggressomes. These findings indicate the existence of transferable signals that target aggregation-prone polypeptides to aggressomes. Human disease. Accumulation of misfolded proteins in proteinaceous inclusions is common to many age-related neurodegenerative diseases, including Parkinson's disease, Alzheimer's disease, Huntington's disease, and amyotrophic lateral sclerosis. In cultured cells, when the production of misfolded proteins exceeds the capacity of the chaperone refolding system and the ubiquitin proteasome degradation pathway, misfolded proteins are actively transported to a cytoplasmic juxtanuclear structure called an aggressome. Whether aggressomes are benevolent or noxious is unknown, but they are of particular interest because of the appearance of similar inclusions in protein deposition diseases. Evidence shows that aggressomes serve a cytoprotective function and are associated with accelerated turnover of mutant proteins. Experiments show that mutant androgen receptor R, the protein responsible for X-linked spinobulbar muscular atrophy, forms insoluble aggregates and is toxic to cultured cells. Mutant R was also found to form aggressomes in a process distinct from aggregation. Molecular and pharmacological interventions were used to disrupt aggressome formation, revealing their cytoprotective function. Aggressome-forming proteins were found to have an accelerated rate of turnover, and this turnover was slowed by inhibition of aggressome formation. Finally, it is shown that aggressome-forming proteins become membrane-bound and associate with lysosomal structures. Together, these findings suggest that aggressomes are cytoprotective, serving as cytoplasmic recruitment centers to facilitate degradation of toxic proteins. Proteins implicated in aggressome formation. Histone deacetylase 6 is the protein that, in the deacetylase adapter protein function, forms Lewy bodies. The regular wild-type protein localized to inclusion bodies. No mutation associated with disease has been linked to this protein. Parkin is the protein that, in the protein ligase function, forms Lewy bodies. The regular wild-type protein localized to inclusion bodies. Parkinson's disease has been linked to this protein when there is a protein. ATAXIN3 is the protein that, in the ubiquitinating enzyme function, forms SCA type 1 and 2 DRPLA intranuclear inclusions. The regular wild-type protein localized to inclusion bodies, SCA type 3, has been linked to this protein when there is a protein. Dynein motor complex is the protein that, in the retrograde microtubule motor function, forms an unknown protein. The regular wild-type protein localized to inclusion bodies, motor neuron degeneration, has been linked to this protein when there is a protein. Ubiquilin-1 is the protein that, in the protein turnover, intracellular trafficking function, forms Lewy bodies and neurofibrillary tangles. The regular wild-type protein localized to inclusion bodies, Alzheimer's disease, potential risk factor, has been linked to this protein when there is a protein. Cystic fibrosis. Cystic fibrosis transmembrane conductance regulator, CFTR, is an inefficiently folded integral membrane protein that is degraded by the cytoplasmic ubiquitin proteasome pathway. Overexpression or inhibition of proteasome activity in transfected human embryonic kidney or Chinese hamster ovary cells leads to the accumulation of stable, high molecular weight, detergent-insoluble, multi-ubiquitinated forms of CFTR. Undegraded CFTR molecules accumulate at a distinct pericentrolar aggressome. Role of the aggresome pathway in cancer. There is emerging evidence that inhibiting the aggresome pathway leads to accumulation of misfolded proteins and apoptosis in tumor cells through autophagy. See also JUNQ and iPod. Further reading. Aggresomes. A cellular response to misfolded proteins. Aggresomes protect cells by enhancing the degradation of toxic polyglutamine-containing protein. Olsman, Ya, Lee, L. Chin, L.S. 2008. Aggresome formation and neurodegenerative diseases. Therapeutic implications. Kermed Chemis Year 15. 1. 47-60. DOI 10.2174 09298670878333306992. PMC 4403008. PMID 18220762. Aggresome formation and neurodegenerative diseases. Therapeutic implications. Role of the aggresome pathway in cancer. Targeting histone deacetylase 6-dependent protein degradation. Zarur, N. Merian, A.B. Gabai, B.L. Sherman, Mai. October 2008. Triggering aggresome formation. Dissecting aggresome targeting and aggregation signals in synfilin 1-inch. J. Bioalchemis Year 283. 41. 27575-84. DOI 10.1074 JBC M8022162000. PMID 18635553.